In this video, I'll be discussing polymerase chain reaction, or PCR. This is a method of amplifying a specific segment of DNA and is one of the ways you learn that DNA replication can be used in the lab. This process is helpful for when you only have a small amount of DNA that you need more of in order to characterize or otherwise study. You need 4 different materials in order to perform PCR. First, you need your original sample of DNA that contains the segment you want to amplify, which will be represented by the black pipe cleaner. Next, you need 2 different primers that are complementary to either side of the segment of the DNA that you want to amplify. These primers are synthetic and purchased elsewhere, so you'll need to know a little bit about your segment of DNA before you amplify it. They'll be represented by the white pipe cleaner. Next, you need a specific type of DNA polymerase called Taq polymerase, represented by the blue block. I'll explain this enzyme in more detail later. And lastly, you need dNTPs, or basically extra nucleotides that will be used to elongate the DNA, which will be represented by the orange pipe cleaner. All of these materials are mixed together in a small test tube and put into a machine that cycles them through multiple temperatures. The first step of PCR is heating everything up to 95 degrees Celsius. This is hot enough to break the hydrogen bonds in your sample of DNA and separate the 2 strands from each other. Next, you cool the mixture down to 60 degrees Celsius to anneal or attach the primers to either side of the segment of DNA you want to amplify. You can see that the top primer starts at the right and will elongate toward the left, while the bottom primer starts at the left and will elongate toward the right. The result is that the segment between, including the primers, will be amplified. Unlike normal DNA replication, these primers will not be removed and will be incorporated into the new DNA strands. Lastly, you heat the mixture up to 72 degrees Celsius, which is the ideal temperature for Taq polymerase to function. Taq polymerase then comes in and elongates the new DNA strands from the primers, using the dNTPs or nucleotides floating around in the mixture. The result is 2 copies of your DNA strand from your original one, similar to the result of normal DNA replication. You can repeat this whole process with the 2 new copies of your DNA segment to make even more copies. First, you heat everything up to break the hydrogen bonds in the 2 copies, creating 4 single strands of DNA. Then, you cool the mixture down to anneal the primers to the parts of the DNA segment that they're complementary to. Lastly, you heat the mixture back up, so Taq polymerase can elongate the DNA from those primers, using the dNTPs in the mixture. This process is repeated many times, creating an exponential number of copies of your desired segment of DNA. Now let's take a closer look at Taq polymerase. Taq polymerase differs from DNA polymerase III, which is the DNA polymerase used in normal DNA replication in prokaryotes, in 2 important ways. Taq polymerase has a higher error rate than DNA polymerase III due to the fact that it has no 3' to 5' exonuclease activity, or basically it can't go backwards while elongating DNA to fix any mistakes it just made, the way that DNA polymerase III can. The reason Taq polymerase is used in PCR is because it works well at the high temperatures that PCR occurs at. In contrast, DNA polymerase III would denature and stop working in the same temperature of environment. The big takeaways from this are that 1. PCR is a method of amplifying one smaller segment of a larger DNA sample. 2. You have to already know something about the sequence of the segment you want to amplify in order to purchase your synthetic primers, so you couldn't use PCR on a completely unknown sequence or to identify an unknown sequence. And 3. You're more likely to get errors from PCR because Taq polymerase doesn't have the 3' to 5' exonuclease activity that lets it fix errors the way that DNA polymerase III does. This video is just a quick overview of PCR. Please review your notes, Panopto recordings, or other resources on ELMS for more details on this process, and good luck on your exam.